Welcome to the University of Hawaii Maui College Water Quality Lab. So in this lab, we service a bunch of different community organizations who measure coastal water quality, and then we have a number of different grants that allow us to go and measure water quality around the island. One of our main instruments is right here. It's a really expensive instrument. It's about $100,000, and it's called a SEAL AA3. So this instrument measures nutrients in seawater and in freshwater and in wastewater. So here is the location of the sampler. So we have all of these different tubes that we can put our samples in, and then the sampler picks up the water from each of the different tubes and runs it through these pumps here, and then it runs it through these different modules. So this first module measures nitrate, this measures nitrite, silicate, which is an indicator of freshwater, phosphorus, and ammonia. So nitrates, phosphorus, and ammonia, if the concentrations of those nutrients are high, that means there's probably some influence from wastewater or some fertilizers that are getting into either the freshwater or the ocean. These are called colorimeters. So they shine light through the sample to tell us how much nutrients are in the sample. The way that this all works is through something called colorimetry. The samples are injected into the system, and we're also putting different reagents that turn the nutrients color. So for example, if we have a sample and we put our nitrate reagents in, so if there's a lot of nitrate in the sample, when we inject our nitrate reagents, then the sample will turn pinkish purplish. So the more pink that the sample turns, then the more nitrate is in the sample. When we inject our phosphorus reagents or our silica reagents into the sample, then the sample will turn like a bluish color. So the more blue, then the more silica is in the reagent. And then we turn that color into a concentration to tell us exactly the quantity of nutrients in the seawater or the freshwater. So that's how this big colorimeter works. Here are called an IDEX system, and it measures E. coli in water. So E. coli is an indicator of fecal bacteria or fecal coliforms. So if there's a lot of E. coli in the water, that means it's really dirty, and sometimes E. coli can be harmful to human health. So a lot of times people will monitor for E. coli in like the streams or in our oceans. To measure E. coli, we first would collect the sample in this little container here, and there's a little line that shows 100 milliliters, so we would fill the sample up to 100 mils. Then we would add food, a certain kind of food to the sample, and that would help the E. coli to grow. Then we pour the sample into these trays, and we place the tray into what's called an incubator, which is kind of like an oven. So it heats up the sample, and we leave it in that incubator for about 18 hours. So this is the incubator that we put our wells or our trays into so that they can kind of cook overnight. So we just open the oven and we place the well or the tray inside of the incubator. Then we close it. We leave the tray in the incubator for 18 hours, and then we come back the next day, pull our sample out, and you can see that it's turned yellow, which means that there's coliforms in the sample. And then we take it to our fluorescent light to determine how much E. coli is in the sample. You can see that many of the different wells have turned yellow, that means it's positive for different types of bacteria. And then we want to know how much E. coli is in these samples. So to do that, we have to place it under a fluorescent light. And the wells that are fluorescing means that the sample is positive for E. coli. Then we count up those number of wells to come up with how much E. coli is in the sample. So to find out if the sample is fluorescing, we put it under this fluorescing light here. So you can see that all of the wells that are fluorescing that bluish color are positive for E. coli.
So he leo mahalo kia ia o ko pakahia pau no ka hui ana no ka ho'ona nea pu ana me mākou no ka ho'oka ike no ka ho'olohe ana I ka ike Hawaii o kia makumu. Mahalo nui for all of you tuning in with us throughout these past days and really, engaging in the ike that has been shared from all these wonderful mentor and kumu of Maui, of Hawaii. He leo mahalo keia, again, mahalo to Ka'ehu, again, mahalo to all the people who shared and engaged with us in our keiki and our community. And again, this is an encouragement to all you folks to find that space in your own community to learn, to engage, to share, to create relationships with Aina and with people and learn more and share more in the culture in the Aina, and I'm so happy to be able to share with you folks today and throughout these past days. So mahalo piha ia o ko pakahia pau o wau no o ka hakuriti kamara ma ka ehu a hui ho launa pu ho kako I ke kahila a hui ho.